Hopefully everybody can hear me. This is my conflict of interest slide. It does take a village to take ideas from a very primitive stage, such as the stage that I typically am involved, and get them actually out into the real world. And I'm indebted to these companies, and I will say nice things about them. For those of you who want to use your smartphone instead of listening to my lecture, I would recommend, this is a distraction for you, pged.org, where you can put your dot on the map here. These are all the sites where there are genomically literate individuals, and this is my wife teaching a class everywhere she goes. She, in addition to being an amazing researcher, she teaches these classes on genetics. So hopefully you won't be disappointed by a survey. I found that this tends to work. It's rather like a tour, but there's articles behind all of this, and I haven't found it useful to go through the articles. Many of these are unpublished, but it's the same idea. Anyway, these are topics: DNA as a material, an in situ method, an in situ sequencing method, and then this open cohort that we just heard about, then CRISPR, which probably needs no introduction, and then the BRAIN initiative, which may also. But so DNA as a material, I'm going to give 3 quick examples, which is the super resolution example, and then a drug delivery, and then information. This is just kind of a small experiment that I did with Peng Yin and William Shi, who the 3 of us, and Pam Silver as well, constitute the DNA nanostructure component of Harvard. And 3 of us, 3 labs, collaborated on making these long rods that allow you to barcode literally in multiple colors at high resolution. So there's about 42 nanometers between these dots, the closest spaced dots. So this is way beyond the resolution of fluorescence microscope. And unlike every previous method for super resolution, the blinking of the fluorophores, and I'm not going to go into the whole physics of super resolution, but the blinking of the fluorophores is here regulated by the binding and non-binding of fluorescently labeled oligonucleotides to the barcode. So the labeling itself provides a neat way of doing flickering for those who are interested in super resolution. Now we call these DNA nanorobots. They are robots only in the sense they have 3 of the key components of robots, which is sensors, logic, and actuators. If you only have 2 of those, you're a machine rather than a robot. And here what they do is they will sense either immune cells or cancer cells based on 2 aptamers, single-strand DNAs that fold up and recognize surface proteins of T cells or cancer cells. And then only if you have an and both aptamers are activated, that makes it the logic, will it open up and deliver the antibodies which are either modulatory or killing, in this case killing. And you can see this little animation is showing it opening up when it binds, the little blue aptamers bind. And here's some electron microscopy showing that where we replace the antibodies with gold nanoparticles, showing that what we designed is what we got. And DNA nanostructures actually are much easier than protein design, which we also do. Pretty much all of these things work without much ado, and undergraduates do it in a summer competition called Biomat. So that's example number 2 of DNA nanostructures we think are fairly useful. Most of the history of DNA nanostructures has been toys, but these 2 we think are potentially useful. And here's another one, and we don't know because it's still early days. This is the Colbert that was earlier mentioned. And here he is hawking my book. We made it to number 57 on Amazon due to Stephen. And we also encoded this book, all 5.2 million bits, into zeros and ones, from zeros and ones to A, C, G, and T. So all books these days are zeros and ones at some point or another. And this is what we call archival storage. It's not like your thumb drive where you're just like fiddling with it all day. It's archival storage. And it has the advantage for that. It's a very specialized field, although it's a multi-billion-dollar business that most of you don't know about. But DNA has the advantage over typical archival storage, which you have to replace every 3 years or so, in that this has a 700,000-year test run, which is better than most other storage media. And we can store about 200 exabytes per gram. So we can fit a zettabyte in the size of a thumb, where an exabyte is 8 times 10 to the 18th bits. or 10 to the 18th bytes. And the way we did this, I mean, we'll come to a moment in exactly the chemistry and instrumentation, but this is the landscape where we have in blue kind of standard consumer storage ranging from the old-fashioned CDs up to modern flash memory and hard drives. So we're getting up into the, this is log 10 on the y-axis and for the density, and then production scale on the x-axis kind of gives you some idea of how big the market is, how cheap it is, and in other words, you don't go out to lots of bits unless it's inexpensive. And then purple are a bunch of physics-based methods which are hard to scale into consumer markets because they involve things like liquid helium, which most people don't want to carry around liquid helium. And then in red are some biologicals. This is where we bumped up essentially 6 orders of magnitude, almost 7 or 8, relative to most storage media in terms of density. And then this year we have some funding from one of the archival companies to increase it by 10,000 fold, so 10,000 fold in 1 year in terms in this axis of cost. So how do we, and this, by the way, this was published in Science along with Sri Kasari was the professor in this case, my postdoc pretending to be professor and me pretending to be postdoc. I did this mostly with my own hands. So you can see why I'm giving a little extra time. So this is the two main, there are many different methods that we worked on with these companies from the very beginning. Inkjet and this photolithography, which actually originated here in Madison. And we had great, I would dedicate this slide to Franco Serino. And anyway, these are two methods which are essentially based on analogous methods in the non-biological world. And here you have four different phosphoramidite chemicals, G, A, T, and C. And here you have their flow through in a flow cell one at a time, and where you can get either photo deprotection. You can get up to 6 million oligos per chip, and the chips can be as little as $200. So this is really inexpensive. And this brings us to an interesting topic, which is why is everything so inexpensive? So as many of you may know, that was all about writing genomes. We have to read them as well, and we read everything we've written. And this is an example of Moore's law curve, which is pretty aggressive. It's 1.5-fold per year, multiplicative, so these are factors of 10 on the y-axis. And if you predicted how long it would take to go from the $3 billion genome to an affordable one, meaning around $1,000, which is right now we're at about $1,000 cost, slightly higher price, and it was predicted to take 6 decades at Moore's law rates, like electronics, but it actually only took 6 years. So don't believe it. Don't feel limited by very long-term trends, I guess is the lesson here. You can't say whether it'll take longer or shorter. Okay, and that's for cost. But the reading is not just about cost. You could reduce cost by making worse product. But it turns out the quality is improving exponentially as well. And just like the previous exponent, the exponent has increased, at least in my limited analysis here. So it's sort of in the 0.3 to 3% range at the beginning of the genome project, and now it's around 10 to the minus 7th, as we'll see in just a moment. That's 1 error in 10 million. And the haplotype phase length, which we'll also see in a moment, is how long you can read where you know that it came from the mother or the father in the case of a diploid human, let's say. Okay, so exponentially improving cost and quality. So what have we done for you recently? Say, what's the unpublished sequencing? So first I have to introduce this super one way of getting super high accuracy, and it's relevant. We originally developed this, we meaning Rob Mitra in the 90s, and then Jay Shindur in 2005, and then with Roddy Dramatic and his colleagues at Complete Genomics, with a series of papers that resulted in the Complete Genomics system, which is sequencing by ligation, but we also helped with the sequencing by polymerase synthesis. And here this is an ordered array. Most of the other methods are unordered. They're Poisson limited, they're kind of random, but this is kind of pretty. It shows that each of these dots was microfabricated, which is coarse, and then a single molecule, albeit a rolling circle amplified molecule, completely occupies each dot. So the dot might be 200 to 300 nanometers, but they're each completely dominated by one molecule, and hence you get these pure colors, you know, yellow, magenta, blue, and green, and you just don't get any bluish green or yellow magentas. That beats the Poisson distribution, and it also, in combination with another method, gives us this really high accuracy. So you dilute 10 cells of genomes, they're kind of broken into the 200 kb range, into 384 well plates, and now the probability of finding mom and dad's genes in the same well is very low. It's about 1/10 of a genome. And then you amplify and sequence these by this ligation method in the previous slide. And you can now tell, you can get the haplotype phase, which is clinically significant because let's say you have good alleles G on a particular gene. So this is one gene, 2 mutations. And the paternal chromosome gene had 2 Cs as alleles. Or you could have those same 2 Cs, if you kind of lose track of where you are as you're sequencing along, because everything's very similar, then you could have a C from mom and a C from dad, which would mean you have 0 functional copies. So 1 functional copy versus 0 functional copies should matter to you if you're going to get your genome sequenced in the physician. Now, oh, by the way, just audience participation. How many people here have their genome sequenced? Okay. We got some work to do. Okay. So that was done with 10 cells, not because we were showing off, but because 10 cells is the right number of dilution so that you get enough copies so that you get high accuracy, and you get the separation that you need for getting distinguishing the maternal and paternal copy, and for that matter, distinguishing other homologs in the genome. But we can go to single cell, and indeed we can go to subcellular, and we kind of skip straight to there because there's a lot of interesting context in transcriptomes. Now we're switching from DNA to RNA, where a cell is not a homogeneous object. It actually varies down an axon or from one side to another of a secretory cell. And so we want 3-dimensional information in the context of multicellular. And typically in cell biology, we feel limited by the number of colors. They overlap in a spectral sense, and we're limited to about 4 colors, practically speaking. But if we can reuse those 4 colors without really moving, so that we can find each pixel again, we essentially get where n is the number of cycles of reuse of the colors, and here cycles, we're calling them next generation sequencing cycles, then we get 4 to the n colors. And that blows up very quickly into a very wonderful large number of colors. So how do we do this? How do we get 3D imaging, 4 to the n colors, where n might be 30 or so at subcellular resolution? Limited only by the resolution of the light microscope, which we've already shown is sort of getting into the 10 nanometer range. So this is, we don't need to drill down on all the gory detail of the method, but basically we take a fixed specimen, we fix with this odd PEG connecting bis succiminal groups. So these will cross-link any amino groups, so there are aminos in the proteins which are fixed. And then as we make cDNA, we add additional amino groups, and they get fixed to the proteins. So you essentially get something that's kind of like a porous gel made up of proteins cross-linking to circular cDNA. So we circularize with circ ligase, and then we very lightly amplify, just enough so that it raises the signal to noise but doesn't make the dots big. So now you have a 3-dimensional array of dots, and each dot can yield a sequence tag. So you have all the advantages of in situ hybridization, but more colors, and you have all the advantages of RNA sequencing, but you haven't lost your place in the cell. Each position of each sequence read is meaningful now rather than random. It's not on a, okay? So this is postdoc Jay Lee and Evan Doherty, mainly, have been pioneering this for many years. Not published yet, but it's in review. So it's rather crowded in the cell. So the cell has about a mean distance, depending on the cell type, of 100 nanometers between any site in the RNA. And so one solution to this is super resolution. So I showed how we can go sort of into the 10 to 40 nanometer range. And molecular stratification and deconvolution I'll show in the next 2 slides. The idea here is you do a subset of the transcripts, and then it will be sparse, and it will be easy for the computer to read the sequence tags. It will be sparse, but you'll be missing some RNAs. You come back in and do another subset, different from the first subset, and now it will also be sparse, but you can add the 2 together, and now you've got higher density in the computer, even though you never had high density in the microscope. So here's an example where you do the subset. You can subset it in a variety of ways, but here you do a subset that ends in A, and here a subset that ends in AC, and here's a subset where it has no preference. You get basically all the priming. And you can see as you go down, it gets more and more sparse and easier to read, and the correlation coefficient gets better and better. And then deconvolution is kind of something you do anyway. I don't mean to be making a big deal about it, but it's something that you computationally do to improve the sharpness of, and this has been a very big impact here. And we have various methods for quantitating the quality of these things. In particular, here's one that appeals easily explained, where we have a database of transcriptome that you're drawing from, or genome, and it tends to be the case that RNAs come from, even though there are some antisense RNAs, it tends to be there's a dominant RNA in every place, and so 97% of what we see is on one strand, the correct strand, if there's a database, and we think there's relatively low noise. Now we'll come back to FISSEQ as a particular application in a moment with the brain. Now what is it about this personal genome project? This is the only open, and it's one of the few integrated, and we argue that it's by n equal 1. So there's big data, which you usually think of as big cohorts. Here we have sort of cohort independent. We're treating each person as if they are n equal 1, even though we have ambitions to have a big cohort. So we have, the idea is not just going from personal genomes to traits in some hugely predictive algorithm, but we leverage the million-fold improvement in cost and quality to fill in some of the blanks in between here. So we can fill in the environmental components. Some of these are either naturally have a sequence tag or can be made to have a sequence tag, like the microbiome is incredibly important for this project, and our immune response to the microbiome tells us, in fact, our immune response to all sorts of environmental factors kind of tells us what our body is thinking about the environment. And so chemicals leave an impression either on the immunome or on the epigenome, that is to say, our RNA and chromatin reflect certain kinds of chemical insults. And we also biobank many different cell types, including B cells and fibroblasts, induced pluripotent stem cells, and some and derivatives thereof. And then with traits. Traits is a big open-ended thing, and this is kind of intended to be like Wikipedia. If there's a trait that's missing there, then you can recommend it, and we'll try to get it in place. This is now spread internationally in US, Canada, UK, and a number of about 5 other countries are in process. And we have approval for fully open access, the world's only open access source for this. And there it goes. And we have an annual meeting on DNA Day, April 25th, and I hope some of you will join us there. Oh, is there anybody here that's a PGP? Huh? It's more than people, then you haven't had your genome sequence. That's a real pity. That's our fault. Okay. So this is the genome's environments and traits. We require 100% on an exam. This is so that we don't tell them later, oh, there's something really bothersome we found in your genome. Do you want to hear about it? That's not the time for them to say, no, we don't want to hear about it, because at that point you've already kind of told them that there's something to hear about. So we try to make sure they know what they're getting into in terms of privacy as well. And then we have stem cells, which we can differentiate into a whole variety of human tissues. Here via the teratoma model, where you get diverse tissues, and here fully in vitro. And some of the traits are quite whole organism. This is a functional magnetic resonance imaging of my brain, actually. These slices are virtual, not actual, as you might have guessed. And this project has now achieved some governmental attraction, in the sense that in an unprecedented collaboration between the National Institutes for Standard Technology and the Food and Drug Administration, they wanted to have genome standards for the world, and they looked around for properly consented individuals and found only one project worldwide, which is ours. And so they're starting with 8 trios from the Personal Genome Project, meaning mother, father, and child. And you can learn about it at genomeinaBottle.org. And also the NIH ENCODE project has now taken on the Personal Genome Project. The ENCODE project is trying to get at all the cis regulatory elements, or every base pair in the genome, finding functions for it. And it's been limited by access to properly consented cell lines. And more beyond consent, you'll see some other advantages, technological ones we have. Now, I'm not going to spend a lot of time on but these are examples of N=1 studies where you either discover a new allele and what you can do with it, or in some cases you rediscover or you apply them. But these are cases where people were not getting what they wanted out of medical care and then found it in their genome. So, you know, like the Volcker children is probably the most famous one, where child was 3 years old getting lots of intestinal surgery, and they did his genome and found out it was an immune function, and so cord blood, and he's fine 5 years later. John Lauerman was one of the PGP early adopters, and he was basically healthy, although he had in his medical records these dark spots on his retina, or scotoma, and he had leg pain that was bad enough that he got hospitalized. And they actually, and this surprised me, they did a genetic test due to his leg, thinking it might be a clotting disorder. They did what kind of a standard genetic test, not even though he had no family history, of a factor 5 Leiden, so that clotting factor polymorphism, and he did not have it. Now, you can't conclude from that he has no genetic risk factors, and indeed when we did his whole genome, we found that he had a JAK2 mutation, which was somatic. It wasn't germline, and that does explain the scotoma and leg problems, and he's taking aspirin, which is not such a bad lifetime price to pay. So but the point of all these is these are sort of N=1 studies, some of them more researchy than clinical, of looking for deleterious alleles and what you can do to solve them. So in each of these cases, the solution is on the far right. But what about the opposite problem? This has been underrepresented, which is, what if you have a rare protective allele rather than... So these are people that manage to live past 110. They're called supercentenarians. And the take home from this is not that if you want to live past 110, you should smoke and drink to excess, but that maybe they have the same kind of environmental problems all of us have, but they have something else that, biological, that's protecting them, that's allowing them to get past all the infections and the chemicals in their environment. So we and others are in the process of sequencing many of these individuals, and you'll have to stay tuned to whether we actually find any protective alleles or not. But there are a few that we can look at as examples, and it also illustrates, more important than just this idea of protective versus deleterious, it illustrates what we can do to move from a correlation-based human genetics research to causality-based. Because in many cases, the N=1 is quite clear. I mean, that is to say, you know, I'm arguing that all of us are N=1, and to do research properly, we need to treat it as such. But there's some cases where it's very clear that in the whole literature, there's only one child who is documented as having a myostatin double null, meaning both maternal and paternal, and there's one child who has a myostatin receptor double null, if I've correctly interpreted the literature. So it's hard to get 10,000 of these children to do a big study, but you can switch to causality, and it actually becomes more convincing than many GWAS studies, because here's 3 different animal models. You don't have to do 3, but in this case there were. This is the double-muscled cow, and then on the right is the myostatin dog, which is, to scale, and this is the intentionally made mouse on the right as well. So that's myostatin double nulls, and that's the top of this list. And here's kind of a list I've collected of rare protective alleles. In addition to myostatin, there's LRP5, which is not a double null, but it's a particular set of, there's a few alleles that result in a heterozygous state of extra strong bones, so strong that they will damage surgical drill bits and saws, and these people are very heavy in water, dense in water. PCSK9 has kind of stimulated the pharmaceutical industry because these people have incredibly low LDL cholesterol levels. In fact, when I first heard about it, I said, can they be alive? And they're not only alive, they have very low coronary artery disease. CCR5 and FET2 are viral receptors, which apparently are fairly normal health-wise in the double null state, but they're abnormal in that they are resistant to HIV and norovirus. Now, APP could be on another slide about deleterious alleles. The reasons on this slide is decode genetics found that although there's many alleles of APP which cause you to be at higher risk for Alzheimer's, there are some that cause you to be a much lower risk, maybe delaying it by 10 or 15 relative to controls. So I want some of that. Okay. Now, how do we get some of that? How do us regular folks get the good alleles, the protective alleles that other people have? So Timothy Ray Brown was one of the first, wasn't exactly gene therapy, but he had leukemia and AIDS, and he went from being one of the most unlucky people of the world to being the luckiest because his donor for fixing the leukemia was not only matched for HLA, but also was a CCR5 double null, which is the co-receptor for the AIDS virus. And this, in part, along with other biological facts, inspired a clinical trial of this company, Sangamo, which I played a very tiny role in many years ago, but not currently involved in. But they made zinc finger fusions with a bacterial endonuclease, FOK1, and only when they're near one of these two sets of 4 fingers each are near one another will they bind well enough that the nuclease will cleave and make a double strand break, and then make a mess of the CCR5 gene, both copies of CCR5. This is now in phase 2 clinical trials and looks quite promising in terms of toxicity and efficacy, although it does work better on heterozygotes than homozygous, and most people in the world are not heterozygous for this. But anyway, I suspect it will get approved by the FDA fairly soon. But there might be something better. This is effective and low toxicity for cleaving targeted genes where you design the zinc fingers to bind to whatever gene you want, wherever you want. These are very challenging to do experimentally. They're very expensive to do. And there is a better way, we think, now, although it's still early days. This is CRISPR for genome engineering, and not just humans, but many other organisms. So we've participated since almost the very dawn of each of these methods in 7 different ways of doing recombination, maybe more, but definitely. There are 2 of them involve RNA, 2 at the top, group 2 introns, which was part of my thesis, and CRISPR, which is more recently, and then there are 5 that are protein-based, and one of them, it's actually DNA plus protein-based. The DNA finds a homology region, while most of these other ones is just the protein. And I'll just quickly summarize these 2 in a couple of slides. The MAGE, or Multiplex Automated Genome Engineering, uses this lambda-red single-strand oligos. It's currently limited in its best form to E. coli, and we have a little bit of a similar method in yeast. And then CRISPR. Okay, but let's talk about what you can do with MAGE rather than exactly how it works. What we've done is applied it genome-wide to the E. coli genome. So each of these little blue dots or bars is a place in the genome where we've engineered a codon, We've engineered every single instance of the stop codon UAG into a stop codon UAA, so 321 changes genome-wide. We did this in little blocks and then we conjugated it in. The important thing was that this was a case of truly genome-wide engineering where we changed a function. We didn't just make a copy of a genome, we engineered it so that, and it wasn't something that was gene engineering, it was really genome-wide. And this had three things that we were aiming at and three things that we found. Metabolic isolation, we can make the organism dependent upon a non-standard amino acid, which is topic number 2. We can put in non-standard amino acids now at very high efficiency without any crosstalk with other machinery in the cell. And finally, we can get genetic isolation so that the DNA, functional DNA, can't go in and out of the cell. And our ultimate goal is to get multivirus resistance. In this case, this one was resistant to T7 virus. But the idea is if you change the genome, the genetic code enough, it'll be resistant to all viruses a priori, all natural viruses. And this was done, Farron Isaacs is now a professor at Yale, or assistant professor at Yale, and Mark LaJoy is a graduate student. And we have 3 science papers, one of them's come out and 2 more this month. Oddly enough, I mean, who would think that you'd get 3 science papers about E. coli codons? But there you go, some things happen. But CRISPR is not MAGE. It can use MAGE as its input, but it is a phenomenon that really traces back to E. coli in 1987, at least. This is the earliest one I can find. But it was basically neglected as a technology. It was essentially junk DNA for a while, and it was finally discovered that it was kind of a bacterial active immunity. And then just in January, it turned into a technology where we and others showed that it could work in humans, and then so there's a few articles. This is actually just a tiny subset of literature now, just a few months later. Humans, mouse, zebrafish, Arabidopsis, Drosophila, C. elegans yeast, and many others. So it seems to work in other people's hands in contrast to many of our other technologies. Not that they don't work, they're just hard and they require companies to get it to work. This doesn't require a company except maybe to make some short oligos. And what happens is these short oligos go into a guide RNA vector. So there's a little bit of constant RNA, and then the variable part here in green. And rather than the protein recognizing the DNA, the protein helps the RNA recognize the DNA. There's a little bit, that's 2 bases that are recognized by the protein. And you make a triplex where you have 2 DNA strands and an RNA strand. And so basically you really only have to program about 15 to 20 base pairs in here. And this was published in Science in January. Prashant Mali is a postdoc, and Luhan Yang just got her PhD a couple of weeks ago. And here this demonstration was doing homologous recombination, but you can also use it for what I showed before with CCR5, where you can make it nulls. But this homologous recombination, we made 2 targets right next to each other, so showing how close together you can get these guide RNAs. And you can do one or the other or both. If you do both, then you typically get a deletion at the correct endpoints. And we compared it to another recent technology that we and others worked on called TALENs. I'm not describing most of these acronyms because they don't really make much sense to me anyway. But anyway, TALENs so it went zinc fingers, TALENs, and CRISPR, and we compare them in the next slide. So here is that case of correcting 2 different guide RNAs near each other, overlapping a TALEN site. So they're as close as they can be to one another and to being at the same point in chromatin, all the chromatin effects and so forth. And this was picked to be a site that's not particularly TALEN insensitive. There are sites that really we cannot design a TALEN to work, but this one is a fairly typical site. And there is a 9 to 22-fold improvement going from TALENs to the guide RNAs. And you can see there's a fairly small range here, about a factor of 2 in efficiency between the 2 different guide RNAs that are near one another. What else can we do with guide RNAs? We can make deletions. So you hold in one end, so you make, this is 2 guide RNAs, so two of these double-strand breaks, where the left end is held fixed and we move the right guide RNA along it. And you can see it's fairly flat in terms of efficiency, sort of in the 10% range for these particular cell types. We can get up to 40, 50%. And this the biggest deletion we've attempted is 83 kb, and it's about as efficient as a 1 kb deletion. So this works over pretty long distances. This is the work of postdoc Susan Byrne, who also did this experiment where she replaced, so most of what we do is in human cells. We're trying to develop gene therapies and so forth. In this particular case, we took the human Thy1, which is an interesting immunological locus, and we made either 1 or 2 double-strand breaks on either side of it in order to replace it with a mouse Thy1. These are really convenient because they're great immunological reagents for faxing. So here you see fax sorting with just the left guide RNA, just the right guide RNA, and with both and none. And you can see what you're looking for is this box here, which goes, it's about 0.1% in the control, 0.5% in just one end. And if you get both ends, it's shot up to 16%. So you can do this, replace little bits of genes, large bits of genes, deletions up to 83 kb, maybe more. And then we're not limited to double-strand breaks. We can make nicks, and we can make variations on this where we've knocked out both nicking, so there's no cleavage whatsoever. So it's completely nuclease minus. And then we can add on domains, either onto the protein or onto the RNA by coupling it to RNA binding domains. So you can put things that modify chromatin or transcriptional activators or repressors on the protein or the RNA. We've done both, and some of this is described in Nature Methods and some in the Nature Biotech. Now, specificity. This, in principle, should come up with the meganucleases, with the MAGE, with the zinc fingers, the talons, but it comes up a lot with CRISPR for some reason, and that's fine. And this is what we've done. In our first paper, we focused on computational and empirical searches, such as the one I showed you, the very small one that I showed you, but we generated computational for the entire human genome, guide RNAs for the whole genome, focusing on sites that were unlikely a priori to be off target. So we took the whole genome and they filtered out the ones that might have a hit elsewhere, and that allowed us to get 90% of the genes and about 40% of the exons within each of those genes. That's one way. There's two ways: computational and empirical. We can also use nicks rather than double-strand breaks. Nicks greatly, 2 nicks are better than 1 double-strand break because each is independent. And we've done that. That's item 3. And then for activation, so this is not cleavage, but activation, we can get not just 2, but multiple sites. And so here's an example of that, where we rigged it in such a way so that each individual activation CRISPR-Cas9 is fairly low level of fold enhancement of expression with the activation domain. But as you get up into 5 and 10 sites, you get up to 30-fold increase over baseline of the transcription of this endogenous gene. And this was published recently in Nature Biotech. And these are where the positions of those natural, so we didn't engineer this, we engineered the guide RNAs to match to natural endogenous sites scattered from up to 2.6 kb upstream from the transcription start site. Okay, and we have other things in the pipeline for improving specificity, but right now it's already adequate for almost everything that we have in mind, except possibly human gene therapy, which we're working on as well. Now this is John Ock. He co-runs my lab with me, and we've had wonderful decades together. And he's the one that designed these 190,000 guide RNAs that we published in January. And as I said, 90% of the human genes and 40% of the exons in those genes. We're also targeting not only these messenger RNAs in exons, but also activation upstream of messenger RNAs, long non-coding RNAs, microRNAs, and these are the number that we've targeted. And we're using those in an interesting project of transdifferentiation, which will be in the next slide, which is we hypothesize that, or we wonder if these are just cell types around the edge, you don't need to read it, but whether any cell type can be turned into any other cell type directly or indirectly, and if so, how many steps does it take, how many factors does it take? This is kind of a general question that one could ask, and it could be fairly hypothesis independent, and I talk about this a little bit in this molecular systems biology opinion piece. As an example of that, here's reprogramming, not too different from what other groups have done in terms of turning iPS cells into neurons, including some groups here in Madison. But in this case, you can see this clump of pluripotent stem cells. They love to sit on top of each other. And then with one of 4 different genes we've found now, this is shown neurogenin 1, but 3 others, we can get them to differentiate into a specific neuronal type, what appears to be, well, by RNA-seq analysis and morphology, to be very specific. And they migrate out from this mass and they make this kind of this loose network of bipolar cells, we call them, where they have just exactly 2 processes coming out from them, almost population-wide. And this is a 98% conversion, maybe more, in merely 4 days. This would, if these are what we think they are in the brain, this would be on the order of 70 or 80 days during normal human development. So you could call this an artifact, or you could call it a technology. Okay, and this is kind of old data, but we have electrolytic activity and synapse formation in these neurons. And the point of all this is not this particular neuronal pathway, but that we're trying to do this systematically using either the CRISPR method or other methods of overproducing or not making nulls. And nulls, something that's hard to do with RNAi. We did RNA sequencing on these, and we can, I'm not going to go through all the details, but the point is we can characterize these cells. And the problem with it is there's not a lot of, there are brain tissue in the databases, but there are not a lot of examples of single cell transcriptomes where you have a pure cell type or a single cell type. So we're hoping the in situ sequencing will help us find out exactly what kinds of neurons these correspond to, if any. It could be that this is not a natural, these are not natural neurons. They don't, but they're interesting in that this is what you can push pluripotent stem cells to become. And so they're telling us something. Now we've worked with Don Ingber's group and William Poo and Kit Parker on organs on chips. All these different organs are represented by model systems where you can have multiple cell types in a mechanically significant environments. For example, here's the lung, which has a air, epithelium, support layer, endothelium, blood, and this behaves much more physiologically than any previous culture condition, rather like having cells on it, just a flat plastic. With Kit Parker, we've done this contractile assay for cardiac tissue, and we can reproduce individual human mutations, and we can test causality, as I mentioned earlier, not just animal models, but in this organ on a chip model. Now I'm just going to end on a quick introduction to this brain innovative neurotechnologies. This was something that my colleagues here listed here and I somehow managed to get onto the political radar. Not that any of us are particularly good at politics, but this was announced by Obama on April 2nd. And I love the acronym that he and his staff came up with, which was Innovative Neurotechnologies, because it's meant to convey something other than a juggernaut, unstoppable mega project, but really to try to bring down costs and improve quality and maybe bring new modalities. So what might those modalities be? We've had a series of papers on this, starting on some of them that predated that announcement in Neuron and Science and ACS Nano and Neuroscience, illustrating the various points. But the one I want to focus on, just to wrap up here, is this idea of integration, or a Rosetta brain, we call it. And the idea is you'd like to have a background information on a behaving animal where you monitor perception and behavior, and then use 4 different kinds of fluorescent in T2 sequencing. Remember, beginning of the talk, fluorescent in situ sequencing. We are developing a way of turning activity, meaning brain activity of various biological activities like calcium or other signaling molecules, into a DNA ticker tape, which we can read with fluorescent in situ sequencing. We've got a little publication on this, but it's by no means reduced to practice. The connectome that we and Tony Zadar are collaborating on, where you can find the connectivity of every neuron to every neuron at the synapses that they make, 1,000 synapses per neuron in some cases. A developmental lineage where we can use CRISPR to make barcodes real time during development. And then finally, the transcriptome, which is what I illustrated already, you can do with fluorescent in situ sequencing. And the idea is not just to read the genome, but to model it and then write, meaning that you change the developmental biology, or you change the neuronal activity in order to test your ideas. And the earlier you do that, the sooner you know how far off you are in your knowledge. So this is the big wake-up call of synthetic biology is, and I'm not going to go through all the bytes that we get for each of these. It's not, in my opinion, not daunting computational, but other people will almost certainly disagree. But these are the different flavors of the Rosetta, the lineage, the connectome, the brain spike potentials or other activity that might be slower, and so on. Here's an example of the Allen Brain Institute. They did a serial section through mouse and human brains looking for transcripts, and they did them one transcript at a time. And so you needed to, you got kind of sparse sampling of the brain, and you needed lots of brains, thousands of brains to cover all the transcripts, but still a terrific resource and to have it out there. But we'd like to repeat this, getting the transcriptome from one brain. It's easier to align the different images. Different brains have different connectivity, and so comparing different brains is problematic, so we'd like to do it all in one. And that's the idea behind the Rosetta brain, where we not only get all the transcripts in one brain, but we get all these other modalities as well. And that's I'm going to conclude on that particular fantasy. And this is people I want to thank on the brain activity map, now renamed BRAIN for Innovative Neurotechnologies. And they're on our papers. Sorry I couldn't spend more time on it. And these are people to thank for the Personal Genome Project and other people on the technology that I mentioned all the way along. And I'll just leave this up while we have a conversation with questions. And I think there's still plenty of time left, but anybody's free to go, I guess, too. Thank you.
You talked about how it's difficult to design tailings for certain targets. What kind of targets?
It seems like they are more methylation sensitive than CRISPRs. Partly they recognize the DNA from the major groove, which is where the 5-methyl-C is. There may be some new amino acid or something that will help with the 5-methyl-C. But there are other sites that don't have any CG dinucleotides in human that essentially, my colleagues at Harvard, Chad Cowan and others, have found you just get zero recombinants out of hundreds of tries, or NHEJ, and we don't know what that is, but and why. And then they did 10 of these, and they did CRISPR side by side. In every case, CRISPR gave like 30 to 80% efficiency for things that we were getting 0% on the Talon. So it's a little early to declare it, but I think that's the way it looks.
So do you think transdifferentiation of ES cells to all tissue types is going to be just one step for just one protein, or do you think neurons are very special?
Well, I hope neurons aren't special, but I'm certainly not naive enough to think that everything's going to be easy. And I even said these may be somewhat artifactual in some states, and we won't know until we really get lots of single-cell transcriptome data. We have reason to believe that it takes more than one factor to, say, for example, to go from fibroblasts to induced pluripotent stem cells, right? And I think to some extent, many of us felt that it's going to take 4 factors to do everything. Well, here's the good news: is there are some cases where it's 4 and some where it's 1. But what we hope is that by finding all the cases where it is 1, we can have that as kind of a starting point, and then we can do 2 factors at a time, sort of semi-systematically, maybe using the 1s as starting points to seed it, and then 3, and then 4, and 5, and go from there. But it's basically it's a sort of a genomic hypothesis independent way of finding out what the developmental landscape is, starting from a cell that clearly wants to go a lot of different directions based on the teratoma assay. Teratoma, it really goes all over the place. Yeah.
I have a question about your work on de-extinction.
Yeah. I didn't mention that.
Yeah, you didn't.
Yeah, but any question is fair, so go ahead. Yeah.
What are the limits currently on de-extinction? I've heard you talk on TED Talk.
Yeah. So the limits.
What all can one attempt to do
Right.
in terms of reviving extinct species?
Okay. So I would say de-extinction is a big topic. We have a whole talk on that, but it's a non-field so far. Although there's a lot of people working on it, talking about it, whole meetings on it. I think the first set of things is bringing back things that are in the freezer. The second set of things are bringing out certain traits which will enable species to survive in the modern world. In other words, address the issues that made them extinct. So, for example, turning elephants into mammoths so they can survive in the tundra, which might be a better place for both them and the tundra. And I could give a whole reason for why that actually could be good for global temperatures. But it may not take changing the entire genome to achieve those ecological goals, and you could get back quite a few traits with a fairly small number of genetic mutations. And that's our hope, is to explore that space initially. As the technology keeps on its exponential curve, by the time we get so that we actually want to change every base pair for some compulsive reason, the technology will be much cheaper. I think that's a project that could have big environmental positives, but we need to be cautious, and we should wait until we should work for the cost down. Yeah. Yes?
So the brain is very rich in different spliceoforms with multiple different genes.
Yeah.
So with physics, you can get information at different spliceoforms.
Right. Yeah, so fortunately, so this is one case where I was obsessing at the beginning about getting poly A adjacent tags, as was the case in ESTs and SAGE early. And fortunately, my people working on this project completely ignored that and just went for randomly primed. And that has the advantage that if you have something blocking the poly A site junction, you're doomed potentially. And while if you do random priming, you get multiple places. You might miss an exon in a particular molecule, but unless that exon is systematically covered with a protein, you shouldn't miss it in the cell as a whole. That said, if you really want to get all these things, we should do more than we're currently doing to get rid of the proteins and to get rid of the secondary structures. When we plot, I didn't show the plot, but we have a plot where we just saturate some certain RNA, or we looked at the saturation of the RNAs, and you can see every place we get a read is where the secondary structure is lower. But it's pretty well covered. The whole transcript is covered, but you can see the spikes. So anyway, it's like RNA-seq, basically. Randomly primed RNA-seq, in principle, you can get splice forms. You can get allelic variations, so if you have a heterozygote position, so you can get allele-specific transcription, which is a cool thing. But it's still a baby technology. Trying to keep expectations low. Yes? You'd like to sequence your genome. Well, would you like to do it with your own hands, or would you like somebody to do it, help you?
Is there a long line?
Well, so there's different ways. So if you wanted to do it quickly and not with your own hands, Illumina offers their clinical service, CLIA approved. Then if you want to participate in the Personal Genome Project, it's getting better, mainly because the international groups are far more efficient than we are, meaning the Boston group. But that's mainly because we're doing so many things that are kind of proof of concept things rather than pushing people through. But there is a line for the Boston part, but if you're qualified for one of the other countries, or one of the other, better yet, one of the other ones that's starting up in the United States. We'll get, we have ways that we're working on that will get the backlog, and there are rumors that there may be ways of getting 1 million done. This is how rumors spread. Okay. Other questions?
With the Personal Genome Project,
Yes.
are you getting very good coverage, better than what is publicly available?
Coverage. What do you
Coverage
mean?
of the sequence.
Oh.
Yeah.
So I'll repeat the question. For the Personal Genome Project, are we getting good coverage of the sequence relative to other public genomes? There are very few public genomes other than ours, meaning at any level. Even the so-called 1,000 genome project does not have 1,000 high-quality genomes. It's a handful of high-quality genomes. We have 200 high-quality genomes that are covered at 40x or better, which is very.
And you get into the repetitive sequences and all that sort of
Well, so going public or private genomes, there's no human genome that's been finished. There's none where there's about 2 to 5% of the genome that's not known, and that section is not something we should dismiss. My colleague Steve McCarroll in my department recently showed that in some of those regions that are not sequenced are many, many human genes that have been making it very hard to interpret their paralogs, which were in the parts of the genome that have been sequenced. They look like SNPs, but in fact they're just a completely different gene, a related gene that's hiding in the centromeres, for example. So one of our ambitions is to sequence through all the centromeres and other gaps, but we haven't done that yet. But the Personal Genome Project are among the highest quality genomes, period, as far as I know. Yeah.
Well, if there aren't any other questions, let's thank George once again.
Thank you.